Hello friends, today we are going to start a new topic, mitosis. It is the process of cell division and the term was given by Walter Fleming in 1882. Mitosis is a Greek word which means thread. In this process, one cell that is known as the mother cell is divided into two daughter cells and these are genetically identical. Here mother cell is divided into two daughter cells and these two daughter cells are genetically identical. In this mitosis, one round of the DNA replication is followed by single round of chromosome separation. Mitosis is a continuous process. It is basically divided into four substages known as prophase, metaphase, anaphase and telophase. Let us discuss about the cell before the start of mitosis. In the late G2 stage, the centrosome has duplicated. There are two copies of centrosome. This centrosome is present in animal cells but it is not present in plant cell. In the plant cell, spindle organizing center is present which has the similar function as centrosome. Nucleolus is present, nuclear membrane is intact. DNA is duplicated. Now the chromosome has two sister chromatids which are attached to each other and this is in the decondensed form. Then comes the first substage of the mitosis, prophase. Prophase is further divided into early prophase and late prophase. Late prophase is also known as prometaphase. First of all, the chromosome starts condensing, nucleolus disappears and centrosome moves little apart and mitotic spindles are formed. Later on, nuclear envelope breaks down, centrosome moves towards the pole and the mitotic spindles starts forming. Some of the microtubules attached to the chromosome at the centromere. This centromere is a specific sequence in the DNA that helps in the separation of the chromosome. Chromosome is now fully condensed. Each chromosome has two sister chromatids which are attached to each other by the cohesin protein. In the metaphase, all the chromosomes are aligned in one single plane in the middle. There are three types of the mitotic spindle or microtubule: kinetochore microtubule, astral microtubule, polar microtubule. We can understand these microtubules from this figure. This is fully condensed chromosome with two sister chromatids. These are attached by the cohesin protein. Construction that is present in the chromosome is known as the centromere. Kinetochore protein is present on both the sides and later on it matures. Microtubules which come and attach to this kinetochore protein is known as kinetochore microtubule. Microtubule which extends from the two poles but are not attached to the chromosome is known as the polar microtubule. These interdigitate each other, that is, these are interlocked. And microtubules which radiates from the centrosome is known as the astral microtubule. When the microtubules attach to the kinetochore of this side tries to move in this direction, kinetochore of this side tries to move in this direction. Thus, they try to move in the opposite direction. As a result of which a tension is created and this tension helps in the alignment of chromosome in one plane along the half or along the middle. Third is anaphase. In this, sister chromatids are separated and move towards the opposite pole. Sister chromatids are held at the centromere by the cohesin protein. This cohesin protein is degraded by the separase, which is ubiquitin cysteine protease. This degrades or cleaves this cohesin protein and sister chromatids are separated. Securine binds to separase and it is inactive. In the anaphase promoting complex is activated. This is ubiquitin ligase. This degrades securine and then the separase is active. This then degrades or cleaves cohesin and the sister chromatids are separated. They start moving towards the opposite poles due to the certain force that is generated. One is due to the shortening of the kinetochore microtubule. Kinetochore microtubule shortens due to the depolymerization, as a result of which pulling force is generated. The two poles also moves further apart because the polar microtubules slide past one another. Pulling force is also exerted by the estral microtubules. As the two poles move further apart, the cell elongates. Telophase. In the telophase, the separated sister chromatids reaches opposite poles. Now these sister chromatids are known as chromosomes. Kinetochore microtubules disappear. Nuclear envelope is reformed. Nucleoli reappear. Now two daughter nuclei are formed. Chromosome starts decondensing and it looks like string. So till now we have discussed how the nucleus divide and two daughter nuclei are formed. Now we will see how the cytoplasm of the cell divides and how the two daughter cells are produced or formed that are genetically identical. The division of the cytoplasm is known as cytokinesis. Let us see the cytokinesis. Cytokinesis, it is the division of cytoplasm. Every mitosis is accompanied by cytokinesis. It starts or begins in Anaphase and ends in Telophase, reaching its completion as next interphase begins. Cytokinesis occurs differently in the animal cells and the plant cells. Let us see them one by one. First of all, the cytokinesis in the animal cell. In the animal cell, cytokinesis takes place by the cleavage. It is guided by the organized bundle of actin filaments. Actin filament and myosin II. These arrange along the circumference of the cell in the cytoplasmic region of the plasma membrane. This forms the contractile ring, and this contractile ring defines the cleavage furrow. This and this both are cleavage furrow. Both the actin filament and myosin II, these slides, and force is generated in the contractile ring. This force helps in the cleavage of the animal cell. When the force is generated, then gradually the cleavage furrow deepens and these meets at the center. Then it is pinched off and two daughter cells are generated. In this way, cytokinesis is accomplished in animal cell. Now let us see the cytokinesis in plant cell. Cytokinesis in plant cell is different from the animal cell because of the presence of the cell wall, which is a rigid structure. So here the cleavage does not take place. So how the cytokinesis takes place? After the formation of the daughter nuclei, polar spindle microtubules comes and assemble along the equatorial plane and forms a cylindrical structure that is known as phragmoplast. Golgi vesicles, which consist of the precursor for the cell wall, also comes and fuses, and they form a early cell plate along the equatorial plane. Then this cell plate merges, and after that Formation of the plasma membrane is completed around the daughter nuclei. Then along the equatorial plane, middle lamella is formed. After the formation of the middle lamella, cell wall material by the process of exocytosis are deposited adjacent to the middle lamella and primary cell wall is formed. After that, the secondary cell wall is formed, and in this way cytokinesis is completed in plant cell. So this is all for mitotic division. In the next video, we will discuss about meiosis that takes place in the sexual life cycle of eukaryotes. If you like this video, please hit the like button, share it and subscribe my channel. Thank you.